Hello everyone. After a long time I'm doing a new video. Okay. Now this time what we'll do is we'll just go through the random topics. Okay. We won't follow any order or series. So just comment whatever part you want to be explained thoroughly. Okay. Now so the first topic what we are taking is the monohybrid and the dihybrid cross. Okay, proposed by Mendel. Now what did Mendel do? That is our first question. Now, Mendel, what he did was he just took a pea plant. Okay. Now, please pardon my drawing. Okay. Now, what he did was he took one pea plant and he took the other pea plant. Now, what he observed was the height of the plant. Okay. Okay, let us take this is a very tall plant. Okay. Some few more leaves. Yeah. Now this is a tall plant. Now this is a short plant. Okay. Now this is one difference which can be seen through naked eyes. Next, the flower color. Okay, it was of some color and this was of some other color. And the pod. Now pea comes in pod. Okay, it does not have any direct seed setting mechanism. Okay, it comes in pod. The pea seeds, okay, are in pod. Now the pod color and the pod shape were different in the two plants. Then the seeds. Now inside pod what are there? There are so many seeds. Okay, now the seed also had a different color and had a different shape also. Now there were seven contrasting characters. One was the flower color, the flower position. The other flower position was the axial position. Okay. The flower position was one, the flower, okay, and the tallness, the length of the plant stem was the other one. The seed shape, the seed color, the pod shape, and the pod color. Okay, these were what the contrasting characters that were being observed by the Mendel. Now he thought that, see, now this plant has some characters and the other plant had some other characters. Now what if I cross them? What will happen next? Now what he did was he did a procedure which is called as emasculation. Now what is emasculation? Okay, let us understand with a new diagram. Okay, now let us take, this is our stigma. Sorry, pardon. Okay, let us take, this is only our stigma. Next comes what? The anther part. Okay, let us take only one anther over here. Okay, our flower is very much poor. It has only one anther. Now these are all the beautiful petals. Next, although it is looking ugly, sorry for that. Now what he did was he did emasculation. What is emasculation? The process in which the male part, see, emasculation, the word itself has e and masculation, that means the removal of masculine part, the removal of the male part. Okay, now what is the male part in the flower? The anthers, of course. So what he did is he took a pair of scissors. Okay, he took a pair of scissors and he cut the male part. Now if we cut this male part, what is left? We are just left with the female part. Now this becomes a completely female flower. Okay, now this is our female part. Supposing this is our female part, and there is what? A male part also. Now there is a petal. Now what he did was he took the pollen grains from this flower and then put it on this female part. Now this is pollination. Now he did this pollination and then he bagged it. Okay, with a polythene bag. Okay, there is a stem also. Okay, don't forget that. He bagged it. Now bagging is what? He is like protecting so that no other anther should come. Now The thing what he was having in his mind is that he took contrasting characters. Now what everyone will explain, I'll continue the same and then I'll explain with the other example. Now what everyone will explain is the tallness, the height. So let us take, this is the tall plant and this is the short plant. Okay, now he takes the anther of the short plant and puts it on the stigma of the tall plant, short plant and the tall plant, if I'm mistaken, sorry. He takes the anther of the short plant and puts it on the stigma of the tall plant. Now what happens is that, of course, germination happens and then all the thing happens, zygote formation, and then the seed setting takes place. Now that seed is again what, sown. Now when the new plant emerges, okay, a new plant emerges. Now what should be the character, whether it should be tall or short or intermediate, because it's a mixture of both of them. Now what Mendel observes is that, it's surprising to everyone, the thing is that he observes the offspring was again a tall plant and it was not the mixture. Now logically if we think, see, one is tall, the other is short, so the product should be what, intermediate, right? That is what our logic says, but this goes out of logic. Now the progeny is what, a tall plant again. Now now Mendel starts to think, how did this happen? Now he comes with a word called as what? He comes with a word called as factor. Now factor, which we know as gene. Okay. Now what is factor and what is gene? Now Mendel called it as factor, but now in the present generation we know it as gene. Now factor or gene is the material in the DNA of an organism which will divide what character will be expressed. Now Mendel will say that, now we took two plants, the first plant was tall, the second plant was short. Now in tall plant there were such factors which were dominant, and in short plant there were such factors which were non-dominant, or also can be said as recessive factors. Okay, now he represented the dominant factors with the capital letters. Let us take capital T, and the recessive or the non-dominant factors as the small letters. Okay, that is small t and small t. You can also take it as capital S and small s and small s. Now this is not required. I'll teach it afterwards if you want. Okay, now we'll go with the general method. Okay, now this is what the factors are. So capital T, small t and small t. Now when we are crossing, okay, crossing happens only when a diploid cell gets converted into a single ploidy cell, right? Now this is a diploid cell. Now the ploidy becomes half, that is only T. Over here also it is half, so it becomes small t when we cross them. Okay, when we cross them we get what, the mixture of this, that is a capital T and a small t. But this means the progeny should be an intermediate. But what did we see in the progeny? We saw that the progeny was tall one. Now he says that we are seeing that the progeny is tall just because there is a dominant factor which will suppress the character of the recessive factor. Okay, this is what is Mendel's monohybrid cross. Now this is just the half of the cross. We'll continue the cross. Okay. Now what he said was the tall and the short plant. Okay, these were what? Parental generation. Okay, I know this is becoming a bit messy. Please try to concentrate. Now this is what? A parental generation. Now these are the gametes of the parental generation. Let me write PG. Okay. Now this is the offspring, that is filial generation. Okay, F1. Okay. Now what he does is that he'll take two F1 plants, okay, or he'll sell a F1 plant. Now, what is F1 plant according to the cross? A capital T and a small t. Okay, now let us take a capital T and a small t and we'll self it. Okay, so it is again a capital T and a small t. Okay, now see over here, the thing is, in the parental generation there was only one gamete. Why? Because both the factors were same. Okay, now when the case is as such, now the in parental generation it was as such like capital T and capital T. Now this is called as what? Homozygous. What? Homozygous. Okay, homozygous. If it is a dominant character, then it is homozygous dominant. If it is a recessive character, then it is homozygous recessive. Now, when there is a case of homozygosity, okay, as such as in the parental generation, capital T and capital T or small t and small t, both are homozygous in nature. When such situation comes, the gamete is only one, because whatever you take this half part or this half part, the gamete remains the same, right? But when the situations as such, the F1 generation comes, that is a capital T and a small t, this is called as what? Heterozygous. Okay, heterozygous condition. Now in heterozygous condition there will be two types of gamete, because see, if we divide it into halves, either capital T can be a gamete or a small t can be a gamete. So we are getting what? Two types of gamete. So over here in F2 generation, sorry, yeah, F1 generation, right? F1 generation का gamete है, so it is FG1. Okay, so the gamete is capital T and small t. Over here also it is same, capital T and small t, right? Now this is becoming a bit fuzzy, so let us go to the next line. Okay, over here, oh sorry, yeah, let us see over here. Now what we had was a capital T, okay, a capital T, a small t crossed with a capital T, a small t. Now the gametes were capital T small t and capital T small t. Okay, now we are crossing this. Now see here, we agree, okay, these two are gametes of the first parent, these two are the gametes of the second parent. Now how do we know that what gamete is forming zygote with what gamete? So let us, okay, example is, there is a situation where this capital T can go and fertilize with the capital T, or there can be a situation where this capital T can go and fertilize with the small t, right? There are probabilities. Now there can be also another probability that this small t can go and fertilize with capital T, or this small t can go and fertilize with a small t, right? So there are how many probabilities? 4 probabilities. So we are going to write all the 4 probabilities. Okay, now capital T with capital T, which forms a capital T, capital T. What is this now? A homozygous tall plant. Okay, good. Next, after this, there is a second situation in which a capital T goes and fertilizes with the small t. So this is what? A capital T and a small t. Now in the third situation, a small t goes and fertilizes with the capital T. That is what? A capital T small t situation. That is again a heterozygous situation. Then again in the last thing, probability, there is a probability of small t fertilizing with a small t. That is what? A small t and a small t. That is a homozygous situation. Now see over here, the interesting part, there are two situations of homozygosity and there are two situations of heterozygosity, right? Now the thing is that this is a tall plant, this is also a tall plant, this is also a tall plant, but this is a short plant. Why such? Because there are two dominant characters over here, so it will be a dominant plant. Here there is one dominant character and one recessive character, but still the plant will be of a dominant character only. Over here also there is one dominant character and another is the recessive character. Over here also the dominant will be overruling, but here there is no dominant character at all. These both are what? Recessive characters. So it is a recessive plant. So that is short plant. Now capital T will give rise to a tall plant. A capital T small t also will give rise to a small plant. Sorry, tall plant. Over here also the same case, the capital T and the small t which will give to a tall plant. And then there is a small t and small t which will give rise to a small plant. Okay. Now this is the monohybrid cross. Completely monohybrid cross. Now the very important point over here is the ratios. Now what are the ratios? The ratios are as such. Now the ratios are as such. The first ratio is 1:2:1, and the second ratio is 3:1. Now what are these ratios called as? One is called as phenotypic ratio and the second one is called as genotypic ratio. Okay, it's regardless of the numbering. Okay. Now, which one is called as phenotypic and which one is called as genotypic? Now, the thing is that you should understand why it is called as phenotypic and genotypic ratio. Phenotypic ratio means the external appearance. Whatever you see in the external appearance, that is called as phenotype. Okay. And genotypic ratios is the genetic formation of the plant. Now, if you see the word genotypic ratio, you will come to know that there is a word which comes as gene. Okay. So genotypic is the genic arrangements. Okay, this is just an example to make you understand. This is not the actual thing. Genotypic ratio is the one which is saying you about the genic formations. Okay, so over here if you are seeing the genic formations, the first one is the homozygous tall. Okay. The second one is the homozygous short, and between two are what? Heterozygous tall. Now see over here, a capital T and a small t, a capital T and a small t. So the genetic formation of these two plants is what? Same. So we are taking these two as one thing. Okay. And now over here, there is only one plant which is having a genetic formation of capital T and capital T. So we are taking this as one over here. And there is only one plant which is having genetic formation of a small t and a small t. So we are taking this as over here. So now this is 1:2:1. This is what? Genotypic ratio. Okay. I'm not having space over here. Okay. This is what? Genotypic ratio. Now there is the second ratio which says that it is 3:1. Now, of course, this is a phenotypic ratio because you know there are only two types of ratios. Very intelligent, huh? Okay. Pardon for my handwriting. Okay, sorry for that. Now that's 3:1. How is that 3:1? See, they are saying about what? Phenotypic ratio. Pheno means what? The external appearance. How does it look to us? Now, see the first three plants. Okay, what are this? All the three are what? Tall plants. So that is 3 in the ratio, and this is the only plant which is what? Short. So this is the 1 in the ratio. So that comes to the ratio of 3:1. That is what? Phenotypic ratio. So by here we complete the first part of our video, that is what? The monohybrid cross.